Tardigrades, those tiny creatures that even though we can't see them are true legends of survival. You've probably heard of them or seen them before, and if not, I guarantee that by the end of this video, you'll be as fascinated as I am. No matter how hard we try, we just can't kill them. Think that sounds like an exaggeration? Well, listen to this. For decades, scientists from around the world have subjected tardigrades to extreme tests that would make any other living thing shudder. They've been shot out of cannons at speeds of 900 meters per second, withstood the coldest temperatures in the universe, and even been boiled at 150 degrees Celsius. And if that's not impressive enough, they've survived pressures 400 times higher than what we experience in Earth's atmosphere, have been frozen for over 30 years, and just to top it off, they've been launched into space and bombarded with cosmic radiation. And still, guess what? They're completely unfazed. So what is it about these adorable microorganisms that makes them the ultimate survivors on the planet? The secret lies in their biology, and while this was a mystery for a long time, a recent study published just last month seems to have uncovered the answer. What we've learned might just change the future of humanity. If we could adapt what makes tardigrades so resilient to our own biology, we might prevent diseases like cancer, survive in environments with weak or no atmosphere, like on the moon or Mars, travel through deep space without worrying about radiation, and who knows, maybe we could even survive in cryosleep just like in science fiction movies. Tardigrades might be the toughest survivors on the planet, but the story of how we came to know these tiny superheroes is just as fascinating as the extremes they've endured. Ready to dive in? It all began in 1773 when German biologist Johann August Ephraim Gozzi, peering through a microscope, saw something truly unusual. Unlike other microscopic animals he'd studied, which swam, squirmed, or beat their cilia to move, he observed a tiny wrinkled creature slowly making its way across a microalga, using 8 stubby little legs, each tipped with small claws. With a gasp of amazement, and perhaps sensing how this creature would captivate future generations, Gozzi decided to name his discovery the water bear, a name that, nearly 300 years later, is just as charming as it was back then. Can you imagine a creature so tiny it could fit between the lines of a fingerprint? Well, that's exactly what a tardigrade is, but it wasn't until 3 years later, in 1776, that Italian naturalist and biologist Lazzaro Spallanzani gave them their official name, tardigrada, meaning slow walker, based on their unique leisurely way of moving. Although, let's be honest, that might not have been the best marketing choice by the Italians, right? In 1842, tardigrades once again caught the attention of scientists when French zoologist and agronomist Louis Doyer decided to study these microscopic creatures in their natural habitat, moist mosses and lichens. What he discovered was astonishing. One day, while observing the tardigrades under a microscope, he accidentally allowed their habitat to dry out under the heat of the lamp. He watched as the tiny water bears, which had been moving about curiously just moments before, began to slow down, as if entering a state of suspended animation. When he returned a few hours later, to his surprise, they seemed to have disappeared, leaving only small dark remnants that he initially thought were their dead bodies. Intrigued, he repeated the experiment and noticed that, in the absence of water, the tardigrades shrank into tiny, wrinkled balls, a transformation later known as a ton. In this state, all their movement ceased, and they appeared completely dead. However, when he rehydrated them, something incredible happened. The tardigrades slowly came back to life as if nothing had happened. This was impossible for other organisms, as extreme dehydration usually leads to cellular collapse and irreparable damage. But Doyer found that he could keep the tardigrades in this state for days, even months, and they would still revive after being rehydrated. Before we continue, if you like what you are watching, make sure to hit that subscribe button and turn on notifications so you don't miss any of our future content. It really helps support the channel and keeps us bringing you more quality content videos like this one. Now, let's get back to it. Doyer documented one of the first cases of cryptobiosis. Crypto means hidden, and biosis means life. This physiological state reduces metabolic activity to nearly undetectable levels, effectively making them the smallest zombies in the world. In this state, the metabolism of tardigrades slows down to 0.01% of its normal rate, which is 10,000 times slower than usual. To put that in perspective, if you consume about 2,000 calories a day, in this state you would only need 0.02 calories a day to survive. That's barely a tenth of a McDonald's French fry, or about 10 grains of rice daily. However, while shrinking into a small dehydrated ball is impressive, it still doesn't explain how tardigrades can survive lethal doses of radiation, extreme dehydration, or the cellular damage caused when ice crystals form inside their cells. What really happens inside these tiny survivors? In 2017, a team of researchers led by Thomas Boothby at the University of North Carolina set out to investigate how tardigrades managed to survive in conditions so extreme that they would be fatal for virtually any other form of life on Earth. The main challenge in extreme environments like the vacuum of space or ultra-cold temperatures is water, which makes up about 60 to 80% of most organisms. Water is the essential solvent for biological processes, but when temperatures drop below freezing, it becomes a relentless enemy. When water freezes, it expands by about 9%, which can cause ice crystals forming inside cells to rupture their membranes or damage internal structures. Additionally, low temperatures can make the cell membrane brittle, leading to breaks in the DNA. Because of this, nature has developed mechanisms to avoid freezing at all costs. While we can freeze and thaw some individual cells, like eggs or sperm, with minimal damage, we unfortunately can't do the same with whole bodies, despite what conspiracy theorists may say about Walt Disney's body being secretly frozen beneath Disneyland. Some species have developed antifreeze properties to survive temperatures close to zero, such as the Antarctic ice fish, snow flea, and wood frog. These creatures use natural compounds that lower the freezing point of their bodily fluids, protecting their internal cellular structures. However, these adaptations only allow them to survive slightly below zero temperatures, far from the extremes that tardigrades endure. Tardigrades don't just survive sub-zero conditions or mild dehydration in their ton state, they can withstand temperatures as low as minus 270 degrees Celsius, so cold that atomic movement practically stops. Researchers discovered that when a tardigrade faces extreme conditions, it begins to slowly and deliberately dehydrate, a process that can take several hours. In this state, they can lose up to 97% of their total water content, significantly reducing the likelihood of ice crystals damaging their cells. Additionally, their bodies contract, minimizing their surface area and reducing exposure to environmental dangers. But how do tardigrades manage to avoid cellular collapse due to extreme dehydration? Thomas Boothby and his team became intrigued by the idea that some hidden mechanism allowed these tiny water bears to defy death. To solve this puzzle, they decided to study what happens at the molecular level when tardigrades enter their ton state. What they found was astonishing. Instead of producing typical proteins that fold into specific shapes, they discovered a class of proteins without a fixed structure, floating freely like loose threads within the cell. Under normal conditions, these proteins appeared unremarkable, but when the tardigrade began to dehydrate, these proteins started to slowly transform, solidifying into a glass-like matrix within the tardigrade's cells. Generally, protein damage within a cell occurs when proteins denature due to heat, cold, or mechanical stress. A clear example of this is what happens when you fry an egg. The well-coiled natural proteins break apart and unravel, giving the egg its characteristic opaque white color. The newly discovered proteins in tardigrades, now called tardigrade-specific intrinsically disordered proteins, TDPs, along with a heat-soluble protein identified in 2021, formed a protective gel-like network that prevented the mechanical stress of dehydration or extreme temperatures from damaging cellular proteins. To test their theory, Boothby and his team took the genes responsible for producing these proteins and inserted them into other organisms, specifically yeast and bacteria. These organisms, which typically cannot survive desiccation, suddenly became capable of withstanding extreme drying, just like tardigrades. The TDPs function the same way in these foreign organisms, creating that glass-like protective shield and demonstrating that these proteins were a key component of the tardigrade's survival arsenal. But the story doesn't end there. While the TDPs protect cellular structures, they cannot prevent the ionizing effects of radiation that break DNA. The median lethal dose is approximately 0.6 to 0.9 grays for mice and between 2.5 to 4.5 grays for humans. Gray is the unit of measurement for ionizing radiation, equivalent to the absorption of 1 joule of energy per kilogram of matter. If a person is exposed to about 50 grays of radiation, even for a short time, they will suffer severe convulsions and die within 5 days. However, it has been found that tardigrades can survive not just 50 grays, but up to 4,000 grays. bodies, and they do this without showing significant signs of degradation. In 2024, a team of researchers from the University of North Carolina at Chapel Hill, led by biologist Bob Gold, conducted a study to discover how tardigrade cells could withstand extreme radiation damage. For their experiment, they selected a specific species called Hypsibius exemplaris, known for its resilience. Although, like all living beings, it remains susceptible to DNA damage caused by gamma radiation. In the first phase of the experiment, the tardigrades in their ton state were exposed to intense doses of gamma radiation far beyond what would be lethal for humans. The team then used advanced genomic and proteomic sequencing techniques to study what happens at the molecular level during this damage. What they discovered was extraordinary. Instead of merely resisting radiation and eliminating damaged cells like our bodies do, the exposure to radiation seemed to activate hundreds of genes in the tardigrades, flooding their bodies with DNA repair and protection proteins. During their analysis, two key proteins stood out. The first is DZP, damage suppressor protein, which was found to bind directly to DNA strands and envelop them, acting as a protective barrier that prevents radiation damage. In populations of tardigrades expressing DSUP, there was a significant reduction in DNA strand breaks. The other protein of interest was TARDIS, a newly discovered protein. While DZP helps prevent damage, TARDIS plays a role in stabilizing chromosomes even when they begin to break down under radiation stress. This protein acts like a kind of molecular glue, temporarily reattaching chromosomes just enough for normal repair processes to take place. To test whether this unique ability of tardigrades could be harnessed for human use, the researchers introduced the TARDIS protein into human cells. Researcher Keita Tazu and his team at the University of Tokyo genetically modified human embryonic kidney cells to express both DSUP and TARDIS proteins and then exposed them to radiation. Compared to controls, these cells showed higher survival rates when exposed to X-rays, exhibiting 40% less radiation-induced damage to their DNA. Now, the question worth asking is why? Why did tardigrades develop these extreme genetic capabilities to survive in such hostile environments? The ancestral environment theory suggests that the ancestors of tardigrades may have lived in more extreme environments than their current counterparts, and that the ability to survive these extreme conditions was conserved as an ancestral trait. For instance, in the early history of Earth, conditions were much more volatile, with extreme and frequent fluctuations in temperature, radiation exposure, and water availability. However, why does this survival ability also work in places like the vacuum of space or the coldest temperatures in the universe? To me, this seems more like an unexpected result than an evolutionary adaptation. The evolutionary byproduct theory suggests that the extreme resilience of tardigrades may have evolved as an accidental byproduct of other, more common stresses. For example, drought tolerance is very useful if you live in an environment like moss or lichens, which retain water poorly and tend to dry out and rehydrate multiple times a day. The same mechanism that allows tardigrades to survive desiccation, such as the production of tardigrade-specific intrinsically disordered proteins, TDPs, yes, I said it again, and other molecular defenses, turns out to be exactly the type of protection needed to survive in ultra-cold environments. These dehydration and rehydration processes likely cause DNA damage, so the evolution of a rapid repair mechanism that is frequently activated makes sense, and by chance, it also makes them incredibly effective at repairing radiation-induced damage. Some believe that tardigrades came from space, which I can understand, though I am disappointed they didn't bring more interesting enhancements with them. However, what they might teach us could potentially unlock the future of humanity. As we begin to explore extraterrestrial environments, tardigrades could become a key ally in our journey, with NASA's Artemis missions planning to establish a base on the moon and beyond into deep space. These environments lack Earth's protective atmosphere, exposing astronauts to cosmic radiation, a significant risk that damages DNA over time. This could limit or even hinder our long-term survival in space. Recently, we've covered a technology being developed by NASA that could be the first true force field capable of diverting radiation away from inhabited areas or zones where astronauts live during long-term space missions. However, this would limit excursions outside those areas to short periods. A long-term solution might be to engineer the TARDIS capability of tardigrades into our own genes, in addition to upregulating our own DNA repair mechanisms, allowing damage to be quickly repaired. Although this idea sounds distant, experiments conducted at the University of Tokyo show that this possibility is already on the horizon. That may take time to materialize, but there are also short-term solutions being explored right now. In a concerning 2020 study, researchers discovered a new species of tardigrade capable of surviving intense doses of ultraviolet radiation. Upon further investigation, they found that these tardigrades fluoresced, absorbing shorter UV wavelengths and emitting longer wavelengths, thereby protecting their cells from radiation damage in the process. In a somewhat unsettling twist, the researchers then ground up these tardigrades and applied the tardigrade paste to small worms. With the paste applied, the worms were able to tolerate much higher doses of UV radiation. Could this lead us to develop a tardigrade sunscreen? Perhaps not in the short term, but a synthetic version could be healthier and more effective. Where I've really seen the most interesting work is in the application of tardigrades' unique proteins, such as TDPs and trehalose, to prevent damage to things like organs being transplanted between patients and even vital medications. Many drugs, such as insulin for diabetics or flu vaccines, must be kept constantly cold to avoid degradation. This is not a problem in developed countries, but it becomes a nightmare if, for example, you try to bring medication to a remote village, or if you need to carry an antidote for snake bites while hiking in the woods. Researchers at the University of Wyoming are working to enhance the thermal stability of various medications using the unique proteins of tardigrades to protect these drugs in the same way they protect tardigrade cells, forming a glassy gel that prevents damage. Tardigrades not only occupy an important place in science, but as I've shared throughout this video, they can help us build a future in which humanity benefits from their incredible properties. Discovering creatures like this fascinates me because they show us how we can draw inspiration from the natural world to move toward a better tomorrow. There are still many aspects of tardigrades that I haven't mentioned, but perhaps we'll explore them in a future video. That's all for today. If you enjoyed the video, leave your like and subscribe to support me to create more content. See you soon in the next video.